Very good morning students, this is your Prasanna ma'am from NEET World. Here we are to discuss Gymnosperms from the chapter Plant Kingdom. If you look into the general characteristics of Gymnosperms, they are embryophytic, tracheophytic, archegoniate, phanerogams. Archegoniate is the female gametophyte, phanerogams means flowering plants. They show primitive seed formation, which means seeds without seed coat. Seeds without seed coat, which means naked seeds, which means that beta naked seeds. This is because before fertilization ovules are not enclosed by ovary walls. We know, right? Ovule turns to seed. Ovule turns to seed. Ovary turns to seed coat. Ovary turns to seed coat, or you can also call it as fruit wall. Ovary turns to seed coat or fruit wall. So yes, seeds, ovules are not covered by ovary wall before fertilization. That is the reason seeds are not covered by seed coat or fruit wall post fertilization. Plants are medium sized to tall trees or shrubs, and certain important characters where you need to remember is smallest plant is Zamia from Gymnosperms. Smallest plant is what, Nana? Zamia from Gymnosperms. Largest plant is giant redwood tree, that is Sequoia. Largest plant is what, ma? Giant redwood tree, that is Sequoia. And living fossil from Gymnosperm is Ginkgo, which has fan shaped leaves. Ginkgo which has fan shaped leaves. This act as living fossil. This act as what, beta? Living fossil. Next, if you look into the root, stem and leaf, vegetative characters, root, stem and leaf. Root is, of course, taproot system is present, but then there are no root hairs or root cap on the roots. There are no root hairs and root cap. To this taproot system, there are fungal associations in pinus. In pinus, you will see what, ma? Fungal associations. That is why in pinus the root system is called as mycorrhiza. The roots of pinus called as mycorrhiza because they are associated with fungus. Fungal association with the roots of pinus will increase their surface area, increase the surface area of the roots, thereby they can absorb more water and minerals. And here if you look into the coralloid roots, these are the roots of cycas are in association with blue-green algae, which is also called as cyanobacteria. Blue-green algae, which is also called as what, ma? Cyanobacteria. So what you need to remember over here? Pinus will show mycorrhiza, cycas will show coralloid roots. Mycorrhiza association with fungus, coralloid roots association with blue-green algae. Association with fungus is increasing the surface area and its level of absorption of water and minerals. Association with the roots of cycas, these coralloid roots, BGA is helpful for nitrogen fixation. It is helpful for what, ma? Nitrogen fixation. Next, if you see stem. Stem is of two types again, unbranched and branched. Unbranched stem you will see in cycas, unbranched stem you will see it in cycas, branched stem you will see it in pinus and citrus. Branched stem you will see it in where, beta? Pinus and citrus. Most important to remember, mycorrhizal roots observed in pinus, coralloid roots seen in cycas. Unbranched stem observed in cycas, branched stem you can see in pinus and citrus. Next, if you look into stem anatomy, stem will show eustele wherein xylem and phloem are arranged along with the parenchymatous sheath. What is the eustele, ma? Xylem and phloem are arranged in the form of strands around parenchymatous sheath, or cells, parenchymatous cells. So if you look into the gymnosperm stem, if you see, This is completely parenchymatous tissue on which the xylem and phloem are arranged in this pattern. In which pattern beta? In this pattern. Xylem will always be towards the center in a stem. Xylem will always be towards the center and phloem will be towards the periphery. Phloem will be towards periphery. Xylem and phloem are interrupted by a secondary meristem called as intravesicular cambium. What is it called as? Intravesicular cambium. And in between these two vascular bundles, intervesicular cambium later develops to conduct secondary growth of the stem in gymnosperms. In between what is developed? Intervesicular cambium. This forms a cambial ring and conducts what beta? Secondary growth. So yes, here xylem is towards the center, phloem is towards the periphery, and the cambial ring formation is conducting secondary growth. The cambium which is present in between xylem and phloem is called as intravesicular cambium. Intravesicular cambium. The cambium which is present in between two vascular bundles is called as intervesicular cambium. Intervesicular cambium. This conducts what beta? Secondary growth. So how you can describe the vascular bundles present in the gymnosperms? They are conjoined, where xylem and phloem are on the same radius. Collateral, xylem and phloem, one vascular bundle is beside the other vascular bundle. They are collateral and open, which means there is a cambium present in between xylem and phloem, then you can call it as open vascular bundle. This you will see it in gymnosperms. Clear beta? This you will see in what? Gymnosperms. We will get to know more clear about this when we are discussing anatomy. Clear? Now, vascular bundles in xylem, if you see, xylem is composed of 4 tissues. Xylem and phloem are what? Complex tissues. Xylem is composed of 4 tissues. Those are xylem fibers, xylem parenchyma, and tracheids. And what beta? Tracheids. Vessels are absent in xylem. In phloem, if you see, phloem consists of sieve tubes, phloem fibers and phloem parenchyma. But then companion cells are absent in phloem. Other than companion cells, albuminous cells are present in the phloem. Here in xylem, what is absent ma? Vessels are absent. In phloem, albuminous cells are present, companion cells are absent. Keep it in your mind. Now, secondary growth occurs in the stem and root because they possess cambiums. They possess secondary meristems, that is cambium, and they conduct the secondary growth, which increases the girth and width of a plant. Now, if you see leaves are pinnately compound in cycas. Leaves are what ma? Pinnately compound in cycas, needle-like in conifers. Why they are needle-like in conifers? To reduce the surface area of leaf, thereby to reduce transpiration. Thereby what? To reduce transpiration. And the leaves are fan shaped in ginkgo, which is the living fossil in gymnosperms. Leaves are adapted to withstand extreme temperatures, humidity, wind, and they have thick cuticle and sunken stomata. Thick cuticle layer over the leaf and sunken stomata, which means stomata is present deep inside the leaf, not over the epidermis, not over the surface of epidermis, but embedded inside the leaf, so that they can reduce water loss by minimizing the transpiration. Now, if you look into the heterosporous condition of the leaf, Here also leaves will bear cone like structures, both male cone and female cone. Male cone will be releasing pollen grains and female cone will consist archegonia and which bears egg to conduct the process of fertilization. If you look into bryophytes, bryophytes are homosporous totally. If you look into pteridophytes, first heterosporous condition you will observe in pteridophytes. In gymnosperms, it is totally heterosporous condition. You will have two types of spores, one is microspore and the other is megaspores. Sporophylls, if you see, sporophylls means what? Those leaves which are bearing spores and sporangia, then you will call it as spores. But here spores and sporangia are arranged in the compact cone like structures, both male cone and female cone. They are spirally arranged along the axis in the form of strobili or cones. Clear ma? Now if you look into sporophylls clearly, microsporophylls contain compactly arranged male strobili, microsporangiate or male cone, one and the same. They contain what ma? Male strobili, which is also called as microsporangiate. This is also called as male cone. And if you look into the ploidy, the sporophyte of gymnosperm contain root, stem and leaves. So all the sporophylls are diploid. They are diploid. And microsporophylls again they are diploid. Megasporophylls are also diploid. All the male and female cones diploid. Here microsporangia will bear megaspore mother cells which is diploid. This conduct what beta? Sporic meiosis. They will conduct what? Sporic meiosis. And they will release microspores. They will release what? Microspores. Microspores will develop into male gametophyte. Microspores will develop into what? Male gametophyte which is said to be pollen grains. Microspores develop into male gametophyte which is said to be pollen grains. This is about microsporangiate. If you look into megasporophylls, this consists of female strobili, megasporangiate or seed cone in which they have multiple archegonia. There is nothing like in a seed coat there is only one archegonia. They can have multiple archegonia, 2 to 3 archegonia. And the topmost position through which the pollen grain reaches this cone is called as micropyle. It is called as what ma? Micropyle. And it has nucellus. One of the cell in nucellus gets differentiated to megaspore mother cell. Nucellus is diploid, right? Yes, when seed cone is diploid, everything which is present inside the seed cone is diploid. Archegonia is also diploid. So this nucellus cells which are present inside the seed cone, one of the nucellus cell will get differentiated to megaspore mother cell. Megaspore mother cell again conducts what beta? Sporic meiosis. They conduct sporic meiosis and forms megaspores. How many daughter cells will form at the end of meiosis? 4 daughter cells. Of the 4 daughter cells which are formed, 3 get degenerated and there is only one functional megaspore. There is only what ma? One functional megaspore. Clear beta? Now if you see that one functional megaspore will undergo sequential mitotic divisions. They will undergo what? Sequential mitosis divisions. And they develop into multicellular female gametophytic structure which bears haploid endosperm. Which bears what ma? Haploid endosperms. When megaspore is haploid, conducting mitosis, this develops into multicellular female gametophyte which is haploid. Inside that there is endosperm. One of the cell is differentiated into what? Endosperm. Here endosperm is forming as a pre-fertilization event. Endosperm is forming before fertilization. So this is a pre-fertilization event. That is the reason in gymnosperms, endosperm is what beta? Haploid. In angiosperms, if you see, in angiosperms, endosperm is formed as a result of triple fusion post fertilization event, and the ploidy of endosperm in angiosperms is triploid, whereas in gymnosperm is haploid. Many times asked question, keep it in your mind, fix it in your mind. Clear? Next, Megaspore gives endosperm pre-fertilization event. Now the pollen grain, which is the mature male gametophyte, is mostly travelled through what? Anemophilus, which means travelled through wind. Okay, it is travelled through wind currents, and this reaches the micropylar region, and here the pollen tube is developed to reach the matured egg. It will reach the what? Egg. Through what, ma? Pollen tube. Through what? Pollen tube. Yes. So this is called as siphonogamous oogamy, which means that pollen grain is reaching to egg through the pollen tube, then it is called as siphonogamous oogamy. Here the egg gets fertilized and it results in the formation of diploid zygote. It undergoes mitosis. This undergoes mitosis, forming a diploid embryo, and this is how ovule, complete ovule will turn to seed, that is naked seed. Here ovule is not covered by ovary wall, ovule turns to seed which is naked. Clear, ma? Yes. So in cycas, if you see, in cycas specifically, if you see, they conduct both siphonogamous and pseudogamous oogamy. Both the types of fertilization modes are conducted in cycas, and male gametes here in cycas carried by, they are multiciliated and carried by pollen tube. Okay, they are multiciliated and carried by pollen tube. That is why they can conduct either pseudogamous oogamy or siphonogamous oogamy. And few of the fern characters are present in the cycas. Few of the fern characters, that is pteridophytic characters, are present in cycas. What are they? Cycas will have cercinate vernation of young leaves, which means young leaves will arise from the compact coiled structures. Young leaves arise from what? Compact coiled structures, that is called as cercinate vernation, and the presence of ramanta. Ramanta are hair-like structures which are present all over the plant body, and the presence of archegonia. Archegonia is present both in pteridophytes and gymnosperms, and it is also present in bryophytes. So if you see, there are three classes of gymnosperms beta. They are Cycadopsida, Coniferopsida and Neetum. Cycadopsida include Cycas, Coniferopsida, Pinus and Conifers, Neetopsida, Neetum. Clear? Now, if you look into the life cycle, final slide, that is life cycle of gymnosperms, the mature sporophyte is diploid. Mature sporophyte is diploid, which contain either male cone or female cone. Both are diploid. Male cone will contain megaspore mother cell inside megasporangium. This undergoes sporic meiosis and forms haploid pollen grains. Haploid microspores, they mature to pollen grains. These are haploid. Clear? And from the male gametophyte, they will reach the micropyle, and through the pollen tube they get fertilized. They will fertilize the egg. So here what is the haploid phase? Only the pollen grain. If you look into the female cone, megaspore is haploid, which is forming from megaspore mother cell. This is forming from megaspore mother cell by sporic meiosis again. This megaspore mother cell will undergo sequential mitotic division, and they will develop into a female gametophyte, archegonia, wherein this is haploid, and inside which egg is also haploid. So this undergoes fertilization, forming zygote, embryo, and that embryo again develops into a mature sporophyte. So if you look into the life cycle clearly, where do you find the haploid phases? Only in the pollen grain and only in the female gametophyte. Rest all the life cycle, all the phases of the life cycle are diploid. That is why gymnosperms life cycle is diplontic life cycle. What is the life cycle of gymnosperma? Diplontic life cycle. Gymnosperm life cycle is diplontic life cycle. as majority of the phases in the life cycle are diploid and there is only one phase, that is pollen grain, and a female gametophyte which are haploid, rest all are diploid, that is why it is diplontic life cycle. That is all with gymnosperms. We will come up with a new chapter and new topic in the coming class. Stay tuned. Thank you.